today we will be reviewing concepts and terms associated with different laboratory techniques. So this involves one, chromatography. It also involves gel electrophoresis. There's some DNA sequencing and also molecular cloning. So exciting stuff and also useful to know for the MCAT. So the first one that we're going into today is chromatography. You guys, if you went to David's lecture on Thursday, you probably learned about this. So this would be a quick review. So chromatography, as you know, maybe from Thursday's lecture, is used to separate. Oh, and if everyone can also mute while they're here, thank you. So yes, David's lecture and chromatography is used to separate, different kinds of molecules based on their properties. And so one of the types of chromatography here is column chromatography, which uses a polar stationary phase. And so a common polar column that they use is for silica gel or alumina. And then they also have a nonpolar solvent, which is considered that mobile phase that is the liquid. And so in column chromatography, so I have kind of a visual on here, the nonpolar molecules tend to elute first, and polar molecules usually elute last when they go through this column, because the polar molecules tend to react with a stationary phase, and that causes more of the polar molecules to stick. And so that's why nonpolar, usually it's the first one that comes out. And then we also have ion exchange chromatography, which instead of a polar stationary phase, it uses a charged column, which attracts and binds compounds with an opposite charge. So when the charge is opposite with the charged column, it tends to cause more binding. And if the charges are the same, it causes more repelling. So then the molecules would most likely come out a lot faster. And a salt water mixture is also used in the cases where there is an opposite charge, to cause the column bound components to elute. So for everything to come out. And so when you're separating like a positive charge and negative charge, for example, the one that is the same charge as the column, that would most likely be the first thing that ends up. So here's a diagram of the ion exchange chromatography one type, but the similar charge as what's in the column is the one that's probably going to be eluted out first. And so researchers would collect that first, and then they would put that salt water mixture to take out the ones that were reacting to the charged column, to get that other part of the mixture, other part of the compound that you're putting in the column. So within ion exchange chromatography, there are two types, and one is anion exchange chromatography, where the positively charged column attracts negatively charged molecules. And so I guess the way that I help to think about anion exchange is that anion is attracting that negatively charged molecule, because we kind of know that anion is related to that negative sign in chemistry. So that's how I kind of think of it. And then for cation exchange chromatography, that's when negatively charged, we have a negatively charged column that attracts positively charged molecules. And so cation associated with the positively charged molecules. And so in this example in particular, we actually have a cation exchange chromatography going on, because these positive charges are being stuck to the negatively charged column. Then we also have size exclusion chromatography, also known as gel filtration chromatography, which separates proteins based on size using small pores. And so in this type of chromatography, usually the larger molecules elute first. So here in this diagram, you can kind of see, so these would be the different pores that we're having in the column, and these large particles come out first because they are bigger than those little pores, and so they can't get stuck within them. And so they just kind of pass through all of them and get out first. While the smaller particles can go in this and they tend to get stuck for a longer amount of time. And so then they would come out after those large particles. And then there's also affinity chromatography, which is a little bit more specific. Ligands are covalently attached in a chromatography column. So like you can kind of see here, we have beads containing covalently attached glucose residues in this particular example. And then we're adding the pool of proteins that we're trying to separate, and we're putting it in the column here. And so once these proteins are added, the target molecules that we're trying to get based on the ligand that we're putting in here would be the ones that are binding to the ligand. And so then after that, we know that, all of the ones that are not quite bound are the excess, or the ones that we're not really looking for. And so that would go into this container here. And then after we know that everything else in the column is rinsed to just be the target molecule that we want, then we would go with another rinse that's a little stronger. And so that would cause the protein that we desire to come out. And then we can get that from the separation. Yes. So then the other one is thin layer chromatography, which is something that you guys probably seen quite often in chemistry lab. But it's a sheet of glass, plastic, or aluminum foil that is coated with a thin layer of adsorbent material. Commonly, it's usually silica gel, aluminum oxide, or cellulose. And so like you see in this image here, it's that stationary paper here, that sheet. And then, so sample is added to the stationary phase, and then a solvent is usually put, or it's usually in the beaker where you're dipping that paper into, and solvent gets drawn up the plate. It usually moves upwards. And so that's how you can see the color separation based on the different colors that you put in on the sheet in this particular example. And what happens to be the case for thin layer chromatography is that polar protein samples usually don't migrate very far, kind of similar as we were talking about column chromatography before, where the polar proteins react more with the stationary phase. And then nonpolar proteins usually elute much further because they're not quite reacting with that stationary phase. And usually the RF value is one that is measured when we do thin layer chromatography, because we're trying to see basically the equation for RF value is the migration distance of a substance divided by the migration distance of the solvent front. So we're seeing, it's a measure to see how much each of these different substances migrate over the total amount of solvent, so we can see which proteins migrate farther than the other ones to determine more about its polarity and other properties.